Good afternoon, everybody. This is Mr. Forshee. Today I want to go over kind of a couple clues or kind of a tricks to find the DNA sequence for your project. Remember, the Biotech project will be due sometime before we go out on spring break as we're kind of finishing up the S11 lab. One of the last final things we're going to do is try to figure out how we could fix a possible DNA sequence. So, you're going to be thinking about PCR for cloning, plasmid for cloning, editing DNA, what type of vectors. This is all in that project sheet that I gave you guys. Today, what I'm going to kind of go over is the OMIM. And here you can start to go like OMIM, and you can hit enter. And it's the online Mendelian inheritance of man. And when you double click on this here, it's going to pop up the program. Now, the big thing with looking for your DNA sequence, you can't find it by the name. So I'm going to go and talk to you about Progeria. Now, again, Progeria is a very limited population that actually has this particular disease or mutation. Now, there's 1,400 different types of mutations that can cause this particular disease. And kind of like with your project, you guys just want to focus on maybe one mutation or maybe a couple different mutations depending on you. But really what we need is just one mutation that you plan on fixing. So it could be like a deletion, it could be an insertion, point mutation, frame shift. You guys are going to have to decide on what you're going to focus on. Now, again, when you're doing this, I'm not going to look for progeria, but what I need to know is the gene that is mutated that can cause the condition. So with this particular condition, I know LMNA is the name of the gene that affects this or causes this particular condition. So when I click on this here, it's going to give me a list. Now, anytime I use those keywords, it's going to highlight it for me. So you can see it's not really in the bottom ones, it's in just the first couple. And might have to search around a little bit. Now, if you notice here, some of this is referring to like interacting protein, muscular dystrophy. But let's try the first one here. I know that with the particular condition, A and C are the point mutations that can happen here with the substitution. And this is something that kind of drew my attention. So when I double click on this here, what it's going to do is then going to take me to this page. Now, this page is going to have information for us to look at and try to identify the particular, again, sequence. Now, where it says external links, you guys want to click on here. And if you notice, it says genome, then DNA, then protein. Well, we're interested in finding the sequence for this condition. So I'm going to look at DNA. And if you look over here also, you could see 1Q22 is going to be really the example we want to use for this. Let me get this out of the way here. So clicking on the DNA, I'm going to go to the very first one and click. And what this is going to do, it's going to take me to a page that's going to show me a whole lot of information. Now, again, as you look here, you could see there's a bunch of variants here. Now, the variance is kind of color coding to show you kind of variation to this gene. So you could see alternating codons. Alternating codons is kind of in two different colors. My main thing here is with the purple and the pink and the yellow. And then even with the red here, like a stop gained, slice region, these are all areas that kind of like drew my attention because this is where we could have some of the mutations. So going down here, we can kind of scroll and you could see the purple here. Now, the purple at the top is kind of telling us that's where an area of frame shift can happen. Now, again, each person could be different in terms of what possible mutation they can have. So you guys are going to have to kind of limit to maybe one particular area and showcase that, okay, here's your frame shift. And if it does have a frame shift, how would you correct that? Like what biotechnology tools would you use to study it? and then what type of tool would you use to maybe possibly fix it? Because that's where medicine is heading. I mean, we're now getting to the point of, like Francis Collins was talking, that before we would treat symptoms like cystic fibrosis, but now said for the first time we're really curing some of these conditions by using our skills that are advancing very quickly in biotechnology. So as we scroll down here, I'm just going to show you kind of a region like right here. I'm going to click on this and double click. And what it's going to do, it's going to show me that area. Now, there's a variation, and it kind of gives you the variation. And then, again, you have the forward strand, but it says a frame shift variant. So this is the consequence that can happen. Now, it even tells you what that frame shift can be. And it's a disruption of the transitional reading frame because the number of nucleotides inserted or deleted is not a multiple of 3. So what happens in this area is we sometimes put in a base pair, take out a base pair, and it causes everything to shift over. Now, again, when you're doing this and you're trying to code for a protein, it's going to be difficult for that, protein to be formed because the amino acid sequence is going to change. Now, down below here, you're going to see these single letters. Now, if you did that insulin activity, you notice that for leucine, like you guys probably have learned before, like LEU is how you identify that protein. Researchers don't do that. They're cutting back, and they're going to just give one letter for that particular amino acid. So when you see an L, that like this one right here, that's really kind of looking at that of leucine. Now, what you're going to see also is when you click on this, and I should have done this already. Okay. It's also telling you the position of that. So 981 is the position for that particular DNA sequence. So that's kind of where your mutations are going to be located. And what you're going to do here is you're going to go back towards the top, and you're going to see where it says download sequence. Now, again, this is the cDNA sequence. And again, if you don't know what this is, you can click on this right here, and that will give you some more information about it. Now, kind of save you a little bit of time, I'm going to click on this. And these are the things you can look for. Now, I'm not really concerned about introns. Introns are the non-coding set parts of the DNA. So I'm going to click that off. And, we can get rid of some of these others if we want to. But right now, I'm just going to leave everything in. And what I'm going to do is I'm going to click the download. Now, what happens here, now this is my sequence. So if you notice, here is, again, the sequence for that particular gene. So you can see it's all A's, T's, C's, and G's. Now inside of that is where that mutation can show up. Now when you do this, you can cut and paste this into a document, like a Word document, and then you can do a word search on it. Now I'm not going to show you that part today, but I just want to kind of show you exactly how you can find your DNA sequence for this. And you can see at the top here, it says LMNA 207 in the protein coding. So this is giving us our protein coding section here. And again, we can also use this in our tool alignment that we used in that other program that dealt with the insulin, because it can help to show differences too. So we're going to be using both. Like I said, I'll put out another video, or Mrs. Nacy will put out another video for you guys to kind of show you, how you can do like part 2 of this. But for my class, this is kind of what I was asking you guys to come up with the DNA sequence. And then, like I said, you can then start finding your gene, because this is the correct sequence. There's no frame shift in here. There's no mutation. We're going to have to input that because it's not always going to be the same. Like I said before, it could be a single base pair sometimes, or it could be a couple base pairs. It just depends on what you showcase. Now, again, I just kind of want to stress students, my friends who are in research, I mean, when I talk to them about this, they have whole classes on this particular program. So I'm just introducing it to you. You guys are going to take and run with this, see what you can find. There are some video clips that are online too. So for example, you look at this video tutorials. This is really good to use too. This will kind of showcase, but I think that's kind of getting too much into it. But this is going to give us a starting point. And all we're trying to think here is like, if we had all unlimited resources, we had any lab we can work in, and maybe we had more information on this particular topic, how could I start to try to fix it? And that's where we're going to head. So I'm going to stop it here right now. I will create another video and show you how I put the sequence into a Word document, and from there how you can kind of identify a possible mutation location. And it's just really a word search that you use in Word, and it will find the location of that particular sequence, and then you're going to show me how you can take and edit that sequence. Okay, thanks a lot, guys. Talk to you later. Bye.